Thank you very much for giving me the great opportunity to present my work at the Boku Sustainability Day. In this presentation, I will explain how responsible water consumption for biomanufacturing processes can be achieved. For biomanufacturing, water recycling is very crucial due to high contamination risk of the final product. Therefore, I will further explain the production process of a biopharmaceutical. In the first stage, the upstream processing, bacteria, animal cells, or other host cells which contain the final product are cultivated in a bioreactor under sterile conditions. This happens in a liquid solution where several thousand liters of water are required. In the second stage, the downstream processing, proteins of interest need to be separated from all hazardous cell compartments. Typically, filtration or centrifugation steps are established in this stage. For cleaning purposes, also a large amount of clean water is required. So, the focus of my work lies on analyzing industrial water for biopharmaceuticals, especially focusing on another metric, the process mass intensity, or short, PMI. The PMI is an established metric utilized for the manufacturing of chemicals. The PMI is calculated by dividing the mass of all resources by the amount of final product. Or it quantifies how many kilograms of resources are required in order to produce 1 single kilogram of product. Furthermore, it can be seen that over 90% of the PMI for biopharmaceuticals consist of water. So far so good. However, there are not yet any detectable consequences of the PMI despite the high contribution of water consumption. There is still a long way to establish an environmental metric to the industry, and I show you how we approach an application of the PMI. Typically, when water is consumed in a process, this costs a lot of money. So what happens if we focus on reducing the water consumption of a process? Will there be a change in labor effort? How will yields and waste behave? And can we reduce the overall footprint of a plant? We approach this topic using methods of change management. We evaluate the economic performance of a process, focusing on environmental metrics, and show the responsible people how changing priorities, goals, and process designs are beneficial for the entire industry. Therefore, we work together with facility planners and biomanufacturing companies in an EU-funded project called Next Biofarm, using models of a software called Biosolve in order to maximize the quality of our results and manuscripts. Thus, we could get a good set of data to be precise with our simulation. In the hypothesis of our manuscript, we combine ecological goals and reduced process cost. The big advantage of the PMI, it can be quantified more easily at early stages at research compared to an overall cost evaluation of a process, which is still under development. So we tackle the following SDGs. With our hypothesis, we combine responsible consumption and production with decent work and economic growth. By teaching the industry how to utilize the PMI as an economic indicator, climate action will be the outcome of our research. While developing our strategies of the PMI, we need to consider all relevant parameters of a biotech process. Is water available in the region? What scales need to be considered? Is the PMI applicable for all biopharmaceuticals? Is the facility already existing, or do we want on a green field with our evaluation? For a greenfield analysis, the amount of assumptions increases drastically. Where is the facility built? How much space do I need? What is the ecological footprint of the plant? Is it even necessary, or can't I fit the process in an already existing facility? I will show you how the production process looks like. It starts in a clean room under sterile conditions. The process device needs to be included for sure, but space for media preparation and cleaning purposes needs to be considered as well. The more space is required, the higher the ventilation effort, resulting in a higher cost and higher CO2 emissions. Larger volumes also require more labor effort for processing. Not only processes can be automated, and water consumption is correlated with the relevant cost parameters. By reducing the amount of required water due to process optimization, overall effort reduces as well. As an example, you can see the entire downstream process of a monoclonal antibody in manufacturing scale, where we designed a continuous process in comparison of a batch process. This facility is capable to produce about 266 kg of monoclonal antibody per year, which is a lot. With the available data, we could simulate the production of a small-scale facility for orphan drugs, but also various larger scales of blockbuster products, up to a total scale of 1000 kg antibody per year. For each scenario, we demonstrate that there is a correlation between the PMI and process costs. After the project, our industry partners of the EU project started utilizing developed methods for further research. Furthermore, the data was published and we started to share our experience at the prestigious conference in Europe, the US, and Japan, resulting in further cooperations. In the next step, we worked together with the ACS Green Chemistry Institute, which is a consortium of the biggest pharma companies in the US. They assisted us to further improve the PMI. We developed a novel metric called Varian in order to quantify water-related CO2 emission of a biopharmaceutical. So at the end, we were giving a helping hand to the industry towards a more sustainable future by showing them how the PMI can be utilized. And all of this happens without fearing economic goals cannot be achieved. And with that, I want to thank you for your attention. Stay safe. Thank you very much.